So thank you for the generous introduction. As I said, I'm a clinician scientist. I'm based mainly at the University of Sheffield, but I have a joint appointment at the Medical Research Council in the Gambia, where I have previously spent time full time, but now I travel back and forth and have a group that spans the two institutes. Are my slides advancing? Can I check?
Yes.
Yeah, great. So just a quick summary, I guess, before I launch into the main topic of what our group do. We like to investigate, I guess, how hosts interact with bugs, but within the context of clinical studies. So they're observational cohort studies, clinical vaccine trials, and very soon human challenge studies as well. And our work is divided into two main aspects. One, looking at the host response to infections, as well as vaccines, and the other looking at molecular epidemiology and pathogen detection, and also the interaction between these two things to look at the potential impact that a vaccine may have in a setting that we're working in, because clearly the sort of bugs that are circulating may be different, and often vaccines are made based on decisions in high-income countries. And we work on sort of broadly two main pathogen types, so respiratory viruses and group A Streptococcus, or Strep pyogenes, or Strep A, which is what I'm going to talk to you mostly about. So my talk's divided into three bits. One, I think I don't know how familiar the audience are with Strep A, but I'm going to emphasize, I guess, why I think it's an important topic and the need for a globally relevant vaccine. We do not have one at the moment. And I'll finish with talking about some of the research that our group, wider group, have done at the MRC Gambia in the last 5 years. So Streptococcus pyogenes, it's a gram-positive coccus. The clinicians in the audience will be very familiar with this. The main thing to kind of say is that it is specifically a human pathogen. When I started working on this, I discovered that apparently mice do not have tonsils. So that is one of the reasons that mouse studies are, not a perfect model for studying this. But it's really an incredibly diverse pathogen when it comes to the diseases that it causes. So it can range very much from throat infection, so pharyngitis, so strep throat that many people see, to mild skin and soft tissue infections, to more severe skin and soft tissue infections in the bottom left corner, like necrotizing fasciitis, sepsis, toxin-related conditions. But importantly, in the area I am in now, really, it's the kind of immune sequelae, many sort of weeks, months, years afterwards, both in the heart and in the kidneys, that really form the global burden that this pathogen causes, as I'll come into. So a really wide spectrum of disease. But on that same spectrum, when we're thinking about both transmission, epidemiology, as well as immunity that's generated, really at the sort of left-hand side of this really is that people can also carry Strep pyogenes in the throat and in the skin asymptomatically. And there's lots of questions then about whether some people are resistant and also whether asymptomatic carriage induces immunity, both protective and potentially pathological in terms of the immune sequelae that I mentioned. So on this slide, I guess the two sort of clinical, presentations that, I see and I've seen in the past in the two areas I've worked in clinically. On the left are headlines from a few years ago from the UK, when after the COVID-19 pandemic, we had, huge surges of invasive group A Streptococcus, which I think you probably have seen in your settings and other settings as well, where there's sadly deaths and often of children. And on the right, really, is what I was kind of faced many years ago when I moved to the Gambia for periods of time. And as an adult physician, terrified me seeing children who essentially have signs and symptoms of irreversible heart valve damage from rheumatic heart disease. And this is really very devastating in an area where, the only curative option is valve replacement, and that is not available in this country. So this is something where there's a very high mortality by late teens, early 20s, and also presenting in pregnancy. The importance of strep A in resource limited settings really is through these immune sequelae and rheumatic heart disease. And these are estimates. I mean, I must add, like many things, that the burden of disease for these in many parts of the world are very poorly recorded. But you can see, like many, infections and diseases that have been neglected, that there's higher burden in more resource limited settings. And this is something that one of my clinical fellows pulled out a few years ago, not his work, but just to sort of show the lack of investment that's happened in the past. So this is a plot showing the average research funding compared to the global DALYs as a measure of burden and impact. And you can almost draw a perfect line with the sort of three big HIV/AIDS, TB, and malaria at the top. But you can see that there's a clear outlier there in terms of, until recently at least, funding for rheumatic heart disease. So this is what's supposed to happen. Strep A causes pharyngitis, and often recurrent pharyngitis leads to an acute illness, rheumatic fever, and then in the long term, rheumatic heart disease. And there are thought to be, over 30 million individuals globally affected, and about 300,000 of the annual 500,000 deaths due to strep A thought to be due to rheumatic heart disease. So this sort of paradigm of what happens really dates back to criteria that we use clinically to try and diagnose rheumatic heart disease in terms of an upper respiratory tract site or pharyngitis being the important trigger and driver for these immune sequelae. But there's a slight paradox here in that rheumatic heart disease is virtually eliminated from many high-income countries. But whenever anyone has looked, strep A as a cause of pharyngitis is actually lower in resource-poor settings than it is in high-income countries. So in the last few years, we and many other people who are interested in this have really started thinking about skin infections as being a potential driver of a rheumatic heart disease in lower resource settings. And I'll talk a lot about skin infections during my talk. So I've already said that, strep A probably kills about, 500,000 individuals a year. Why don't we have a vaccine? So some of you may or may not be aware of the history of this, but a lot of the barriers until recently have been regulatory. There was, let's say, a very poorly designed and conducted trial in the 60s that raised fears of higher incidence of acute rheumatic fever in a vaccinated cohort. There was no control group. And this led to an FDA embargo on using any strep A products in bacterial vaccines. And that lasted until 2005. By, of course, which time rheumatic heart disease was eliminated from many high-income countries. So the commercial drivers of producing a vaccine were less. So this fear of autoimmunity is definitely a real one, and certainly something that's, at the heart of vaccine development at the moment. The other thing is available treatment. Thankfully, strep A remains exquisitely sensitive to penicillin, other than very, very rare occurrences. So it's considered that, perhaps we can treat our way out of this with penicillin, but, there's lots of penicillin availability in many settings, and clearly it's not the answer, and there's still a lot of rheumatic heart disease. But I'm going to focus a bit on some of the scientific barriers to this and how some of our work was shaped by these knowledge gaps around what natural immunity looks like, as well as antigenic diversity in settings like the Gambia. So thankfully, things have changed in the last few years. So the World Health Assembly declared that designing a vaccine and tackling the burden of rheumatic fever and heart disease was a priority. And more recently, WHO have got a roadmap with target product profiles and so on for vaccines. And there are organizations through different funding streams like SAVAC, the Strep A Vaccine Global Consortium, that are really driving this, trying to fill knowledge gaps and facilitate vaccine trials. There's 3 at the moment, I think, either in or about to go into phase 1 trials. So it's still very slim in terms of the pipeline, but there are many other in preclinical development. But one thing SAVAC and others have pointed out is that really there's a lot that we don't understand about how natural infection and immunity develop, especially in high burden settings like the Gambia. and that how natural immunity to non-invasive infection develops, because this is what's supposed to drive rheumatic heart disease, leaving aside, invasive disease, and whether carriage is actually protective in immunity, and what the correlates of protection are. So a few years ago, a few of us, while I was still based full time here, decided to kind of set up an interest group in Strep A research, because we thought that we were seeing lots of rheumatic heart disease and we know next to nothing about all of these areas I've listed. So I'll touch on some of them. I'm not going to talk about the rheumatic heart disease screening because that's been led by other colleagues, but I'll mention a bit about these other aspects that I've put in bold. So for those of you who aren't familiar, the Gambia is a very, very small country in West Africa, landlocked apart from the one coast, surrounded by Senegal. And you can see that the age distribution is very different, to where I live in the UK and where most of you live, a median age of 17, a very poor country, and that doesn't fare very well in the Human Development Index, as well. Many individuals, so we work largely in the coastal regions, the studies I'm going to present, which is the most sort of peri-urban and urbanized region where, people live in multi-generational households. And these are some pictures with the field of one of our field teams during one of the Strep A studies. So just to demonstrate what these households are like, these are just some social contact matrices that one of our fellows, Edwin Armitage, put together from a Strep A study, and show the median household size is 7, but the range is very large. So there are some that are collections of small households all living together, up to sort of 37. And these matrices just show the number of individuals that an individual has had contact with in the last day, stratified by age. So on the right, the non-household contacts, you can see a clear pattern, and something we probably see in high-income countries too, where you generally interact with people of your own age. But on the left, you can see this intergenerational mixing that happens, where young children mix with young children, very, very young, before they would go to what a traditional daycare, sort of age is, because they mix within households a lot. But also there's mixing, between old individuals and children. So the first study that we did was a cross-sectional survey of under fives, and this was led by Edwin, one of our fellows. He was actually working on a flu vaccine trial of mine, and we found that although we were trying to follow individuals, children up for influenza-like illness, actually they were coming to see us with lots of skin infections. So we wanted just to see what the burden of skin infections, not just bacterial, scabies and fungal as well, and generate the first microbiology from skin infections in the Gambia that have been done for a long time. So we went door to door in a region called Sakuta, and found that actually with active case finding at a cross-sectional prevalence of 17% of children had some sort of skin infection, and when we cultured those, 50% of them grew group A strep, quite a lot of staff as well. And more recently, we've updated those figures with some PCR data, just showing that actually 80% of those skin or pyoderma skin infections had group A strep in them, which if you go with that intensity of case finding, that means at any one point 14% of children were being exposed in an infection to group A strep, even leaving aside the pharyngitis. So this is a busy slide, but this is some genome sequencing that we did, and Sekou, a talented bioinformatician at the unit here, did. So shown essentially on the y-axis are different geographical clusters in this area, and on the x-axis is time. And each color is a different type of Strep A, and it's typed using the outer surface sort of M protein. And what this essentially shows that there's a huge diversity of types of Strep A causing disease, even in close geographical and temporal proximity. And this surprised us a lot because of the epidemiological framework that we were sampling from. And these are, I think, divided into week blocks. And this is sort of different to what we might see in a classroom in the UK or Canada when we have outbreaks of Strep A. There's clonal expansion. So it's made us think that there's a lot of Strep A around that we're not detecting and that we don't know about. And this diversity in a low-income sort of setting is something that has been seen repeatedly in studies. This is a really nice paper from collaborators of ours showing essentially sort of social determinants of diversity of Strep A. In low-income countries, there's far more diversity of Strep A, which has implications really for how vaccines are designed and whether they're going to be effective in those settings. And I'll come on to talk about some of those. So we wanted to go on to do some epidemiology on Strep A. We wanted to know really what the extent of skin carriage and pharyngeal carriage is, because if there was so much diversity causing disease, there must be more around that people are just carrying and transmitting to each other. And we wanted to establish what the sort of incidence of pharyngitis and skin infection were in a community setting rather than in a clinic. So this is the main study I'm going to talk about called Spy Cats. It was led by Edwin, funded by Wellcome Trust PhD, and Gabrielle was a pediatrician that came from a group in Brussels and helped, and with a very, very large field team there, as you can see. So this was the design. We recruited 44 households and 440 individuals. They were all swabbed routinely, regardless of symptoms, every month with a pharyngeal and a composite skin swab. And then there was, passive to active surveillance of symptoms for throat infections and skin infections. And then if that happened, they were swabbed as well. There were a couple of other nested sub-studies looking for carriage duration and seeing whether if we swabbed more intensely, we would pick up more. I'm not going to talk about those in the interest of time, and I'll just give some headlines from the main incidence data that we generated. So these are just summarized outputs from some Cox hazards models looking at risk factors for in the throat. So compared to individuals 18 and over, you can see that there's a much higher risk of both carriage and pharyngitis in this middle age group of 5 to 11-year-olds, where we actually do see a similar thing in sort of high-income countries as well. But the really striking thing was with skin. So again, the sort of hazards ratios were just so very, very high in the very young children that then gradually sort of came down as you came into adulthood. So as this is sort of summarized as sort of incidence here. So in the columns, so carriage on the left and disease on the right. So pharyngeal carriage and pharyngitis, skin carriage and skin disease. And just to point out, two things to point out from this. One is that there's a slightly different pattern where looks like there's a sort of peak in the sort of 5 to 11-year-olds of what's going on in the throat. But in skin, it's really the very young children that are both carrying more and getting lots of skin infections. But the other thing is really that there's a dramatic reduction as you get to adulthood. And this reduction is really one of the rationales for the fact that we probably do develop some natural immunity, possibly from repeated exposure as we get to adulthood. So that then kind of led to us nesting our sort of other second bit of this program, which was to try and generate some, very unique data on what happens to individuals in this setting in terms of their immunity. So here we wanted to kind of define what happens with age, what happens after people get infected and after they get colonized, but also looking at different antigens. And I'll come on to the kind of relevance of what I've listed there. And we wanted to see that within an observational cohort study, can we kind of define any surrogates of protection to antigens that are now being thought of as candidates in vaccines that are being designed. So Strep A or Strep pyonese vaccine design has taken two broad approaches. One is to focus on M protein. So this is the surface protein I mentioned before. It's highly immunogenic, but it's highly variable. And so there's been some data in 2 or 3 studies suggesting that type-specific antibodies are protective. So if you get an antibody to M1, for example, that then you'll be protected from getting M1 pharyngitis again. But as I've highlighted, that the problem here is that, we found 140 odd different M types in the Gambia, and the most highly valent vaccine at the moment, based on M types, is a 30-valent vaccine. Now, that it's possible that there's cross protection and broader protection, which we can talk about later if anyone's interested. But the other approach really is to look at conserved antigens, and some of them are listed there. So we decided to focus on these because we thought they might be most relevant to the population that we're working in, and we partnered with the Vaccine Institute for Global Health, which is a part of GSK, and decided to look at 4 antigens in their sort of candidate vaccine, but these are also in other vaccines, but other partners are developing, other industry partners are developing as well. So with GVGH, we kind of developed a binding Luminex assay to look at these in our cohorts. And this is a busy slide, but it's essentially an add-on to the prior slide I showed, just to show that as well as all that swabbing, we did some biological sampling, taking serum and dried blood spots at frequent time points before and after events, as well as mucosal data, mucosal samples. So I'm going to focus on the serum and the dried blood spot data today. But this work was led by another clinical fellow funded by the Wellcome Trust, Alex Keeley, but helped very ably by Fatimata Kamara, who's a scientific officer here, who's now gone on to do a PhD with collaborators in Perth. So they tested a huge number of samples, numbers listed in the bottom right-hand corner. So in addition to this cohort, we also then wanted to look at what was happening during birth, because we had some young children, but we weren't designed to look at mother-child sort of transfer. So this is a separate samples from a separate study. And shown here are 5 antigens that we looked at. DNase B, which is often used clinically to look at if someone had been exposed to strep A, and the 4 vaccine antigens. And what's shown here is that firstly that there's pretty good match between maternal serum and cord serum, other than for one antigen, group A carbohydrate, GAC, not shown there, but there was less transfer there. And as once you get to kind of 4 to 6 months, you get sort of waning of maternal antibody to these, which is what we see in other pathogen-specific antibodies as well. But the interesting thing is, in about a quarter of children, we then started seeing boosting between 6 and 11 months, just I think showing what the force of exposure is to this pathogen at a very, very young age, which has implications for, I think, priming for rheumatic heart disease, and also when we might want to think about vaccinating. And then these are data from our baseline samples looking at age and antibody titers to 4 of these antigens. And the summary from this is that we get a very, very rapid rise in antibodies to all of these titers. We get a sort of peak by about age 3 or 4, although there's still a fair amount of heterogeneity. And then it essentially plateaus with potentially some reduction at older ages, but we really didn't have lots of elderly people to really make any strong conclusions about this. So again, I think this has implications for timing of vaccines, but also thinking about the heterogeneity that's there, and whether we can actually improve on natural immunity at any stage, really. So this is a summary of a lot of work done by Alex, looking to see whether we can find any signal of protection from these to antibodies to these antigens, and to 3 of these conserved antigens, adjusting for other factors like age. Alex found that there did seem to be a protective effect. Obviously we don't know specifically if it's due to these, but so they're probably more likely to be a surrogate, and we will only find out through other mechanistic studies and challenge studies we have planned. But it did seem that, at least for the first time in a human cohort, we could see that some suggestion of protection from antibodies to these vaccine antigens. And I mentioned M proteins. We wanted to look at type-specific antibodies as well. And so again, Alex developed a Luminex antibody panel looking at a whole range of different M types. And the summary from this is that, compared to the conserved antigens, the signal was lower. and more heterogeneous, which really I think is expected when really any one person is probably exposed to one different M type rarely. And so, you'd need to be exposed twice to get boosting, whereas the conserved antigens, you would like to get boosted every time you were exposed. And, we did see that there were higher antibody titers to types that we have seen in our genomic data to be more common in this setting. But we wanted to compare these two, and what we were able to do is, because they weren't very well correlated, to put them in the same model. And the summary from this is that it does look like, there may be an independent signal for both, although this was stronger for the conserved antigens. And the sort of the plots in the top just show the probability of infection by antibody titer to the M proteins on the y-axis and the conserved antigens on the x-axis. And you can see at the bottom left, if you got no antibodies to either, you're at highest risk of getting strep A. And the top right, if you've got antibodies to both, you're at lower risk, and gradients in between. So we're excited about these data, and we hope that it suggests that there may be a promise for both approaches. So that's all I have to say, and I'm just going to summarize by saying I hope I've convinced you really that a vaccine for strep pyogenes or strep A suitable for use in areas with the highest burden of rheumatic heart disease is really a priority, and we hope that we see vaccines coming into phase 2, 3 trials in the next few years. So thank you very much for your attention.